joining me today is Professor Hope Rugo, Division Chief of Breast Medical Oncology and Professor of Medical Oncology and Therapeutics Research at City of Hope. Hi, Professor Rugo. Lovely to meet you.
Hi, nice to see you. Thanks for having me.
So we've been asking oncology experts at the meeting how we can ensure that scientific breakthroughs translate into meaningful improvements in patient outcomes globally.
We're in a time of very rapid change, and that's actually fascinating. We can do big data now. We can use computers to try and understand how we could use either gene expression profiles or maybe combine, gene mutations and gene expression and proteomics to really design something where you could understand the better treatment for each tumor type. I think that what we need to do is plan our studies so that we're getting the biomarker results. We have rich translational studies that can then go back and inform us about the optimal way to treat our patients. This is going to really improve outcome.
Yeah, I mean, it sounds fascinating. And I think one of the questions I have for you is when you talk about the clinical trial design, how can we see sort of better collaboration across healthcare much quicker?
Clinical trials give the drugs to patients. They give the opportunity to the patient. So the clinical trials are pretty clear in that part. Doing the translational work, the biomarker data is what's complicated. And a lot of times, for example, a trial will be funded as an investigator-initiated trial or a collaborative trial as the entire trial, but without biomarkers. And the idea is then you apply for separate funding for the biomarkers. So they've always been sort of on the back burner of what's important in a clinical trial. And I do think we need to change that.
My last question to you, I think if you think about healthcare system improvement over the next decade, what success do we ultimately need to look like for patients to sort of start to see this improvement kind of coming through in cancer care more, from a global setting?
In the next decade, how are we bringing this to patients? I think we're already bringing it to patients, and we're on the verge of being able to use ctDNA as a biomarker for early response and potentially improving outcome by changing therapy. We don't know yet. And, looking in the early stage setting at this sort of molecular evidence of residual disease using ctDNA, as the test continues to become more sensitive and hopefully cheaper, because I think the expense is going to be a big issue worldwide, using the continuum to intervene early, that would be interesting. Understanding those who are positive at the start. If they don't clear, we need better treatment options for those patients. And then we could interrogate the ctDNA and the tumor to try and better understand potentially a target that would be result in better outcome and potentially prevent recurrence. There are a whole bunch of new antibody drug conjugates, for example, that are being studied. Could we use this sort of sequencing approach to improve outcome? And these are really exciting areas in the future.
Wow, it's all fascinating. So thank you, Professor Rugo. It's a pleasure to talk to you, and thanks for being in conversation with Real Chemistry today.
Thanks for listening.